All right, let's get right into it. Today, we're going to break down absolutely everything you need to know to master Lab 3 on biological molecules. We'll go over the core ideas, the exact procedures, and how to get ready for that pre-lab quiz.
Feeling maybe a little bit swamped by all the different tests and the steps in the procedure? Hey, don't worry about it. This is your complete guide to walking into that lab feeling totally confident. So here's our roadmap. We're going to start with the most fundamental part of any good experiment, controls. From there, we'll dive into the 4 key tests you'll be using, how you'll actually test your food sample, then we'll get into the science of hydrolysis, and finally wrap it all up with how to nail that pre-lab quiz.
Okay, let's jump in. Before we even think about touching a single chemical, we have to talk about controls. Honestly, if you understand this one concept, you're on your way to getting results you can actually trust in any experiment.
So what exactly are they? Well, a control is basically your baseline. It's your standard of comparison. Your negative control, which is usually just distilled water, is there to prove that your test tube wasn't dirty and to show you what a nothing happened result looks like. Then you have the positive control. For this, you use a substance you know will give you a positive result. This proves that your chemicals are good and your technique is working. Without these two, your results are pretty much just a guess.
Okay, with that foundation set, let's get into the actual tools for this lab, the 4 big biochemical tests you're going to use to figure out what macromolecules are in your food sample.
First up, we've got the Benedict's test. This one is for finding reducing sugars, and that includes all monosaccharides. Now, how does it work? It's what's called an oxidation-reduction reaction. The sugar gets oxidized, the copper ions in the blue Benedict solution get reduced. But, and this is a huge detail for your quiz, this reaction needs a push. It needs a catalyst. You absolutely have to heat it. And here's what you'll see. If your solution just stays that boring blue color of the reagent, that's a negative. But if you see a color change, I mean anything from green for a little bit of sugar all the way to a strong brick red for a whole lot of sugar, that's a positive test.
Next on our list is the Lugol's test for starch. Now, starch is a polysaccharide, which is basically just a super long, coiled-up chain of glucose molecules. And it turns out the iodine in the Lugol's reagent fits perfectly inside that coiled helix structure. It's a really specific fit. If the molecule isn't coiled up in that exact way, the test just won't work. The result you get here is totally unmistakable. A negative result will just look like the yellowish-brown color of the Lugol's iodine itself. But if there's starch in there, you get this really dramatic shift to a deep blue-black color. Seriously, it's one of the easiest results to spot in the entire lab.
Now, for protein, we're going to use the Benedict test. And here's a really important distinction to make. This test does not detect individual amino acids. It specifically reacts with the peptide bonds between the amino acids. What that means is you need a whole chain of them, a protein, to get a positive result. A single amino acid just floating around won't cut it.
This one is pretty straightforward, kind of like the Benedict's test. If it's a negative result, the solution just stays the same blue color as the reagent you put in. A positive result, though, one that tells you protein is present, is a really clear change to a violet or purple color.
And finally, we've got the Sudan IV test, which is for lipids, fats and oils. This test works on a completely different idea, solubility. The rule of thumb in chemistry is like dissolves like. Sudan IV dye is nonpolar, and so are lipids. Water, on the other hand, is polar. So the dye will happily mix in with any lipids, but it wants absolutely nothing to do with the water.
And this gives us a really unique result to look for. In a negative test, that red dye just kind of sits on the wooden stick, not mixing with the water at all. But if there are lipids present, the dye will leave the stick and dissolve into the oil. It creates these super obvious floating red droplets, or you might even see a whole red layer on top.
All right, so now we know the theory behind these 4 tests. Let's get down to business. How are you actually going to use all this to figure out what molecules are in your unknown food sample? Let's walk through the procedure step by step.
So, this flowchart might look a little intense at first, but let's just break it down logically. When we think like a scientist, we think about variables. The independent variable here is the reagent you add. That's the one thing you are changing on purpose for each test. The dependent variable is the result you see, the color change, because it depends on which reagent you used. And your constants are everything you keep the same to make it a fair test, like using the same amount of food and water. So, the procedure is all about being efficient. You prep 2 main tubes, A and B. You take some liquid from A for the Benedict's and Benedict tests. Then you boil what's left in A and B. And why do we boil? Well, boiling breaks open the cell walls, which is super important for releasing any starch or lipids that might be trapped inside. This makes sure your Lugol's and Sudan IV tests are actually accurate.
Okay, let's get through one more key concept before we start talking quiz strategy. Let's talk about hydrolysis. You can think of this as the chemical version of digestion. It's how your body, and how we in the lab, can chop up big molecules into their smaller parts.
The name really tells you everything you need to know. Hydro means water, and lysis means to split. So hydrolysis literally uses a water molecule to break the bonds that hold monomers together. This is exactly how a giant polysaccharide like starch gets broken back down into all its little individual glucose units.
This table right here just captures the power of hydrolysis perfectly. Beforehand, you get really specific results, right? Starch is negative for Benedict's, but positive for Lugol's. Sucrose is negative for both. But after hydrolysis, look how everything flips. The starch and sucrose both get broken down into reducing sugars, so they both suddenly test positive with Benedict's. And because that coiled structure of starch is now destroyed, it tests negative with Lugol's. It just goes to show how one single chemical process can completely change a substance's properties.
We've covered the concepts. We've gone through the procedures. Now for the payoff. Using all this knowledge to walk in there and just ace that pre-lab quiz, locking down those points for your final grade.
Now remember, for this lab, you get to bring in a handwritten index card to the quiz. This is your cheat sheet. So what should you put on it? Exactly this. Write down the positive and negative colors for all 4 of those tests. Make a big note for yourself that Benedict's is the only one that needs heat. Jot down that hydrolysis breaks starch into reducing sugars. Remind yourself that controls are for verification and comparison. And finally, describe that unique positive result for the Sudan IV test, the red droplets, because it's so different from the others. If you have this stuff on your card, you're going to be ready for whatever they throw at you.
So let's end with a quick question, just to get you thinking like a scientist. If your Biuret test turns violet, but your Benedict's test stays blue, even after you heat it up, what can you conclude about the molecular makeup of that food sample? Thinking through little scenarios just like this is the absolute best way to prepare. You've got this.